Oh. Do I need to, oh, there you go. First off, I just want to thank the organizers for this wonderful opportunity to share the work that's being done in my lab. I have to say, I have to apologize. I think I brought the weather from all the way from Portland. It's constantly, it's like 9 months out of the year it's like this. And currently it's really sunny over there, so sorry about that. But anyways, so my lab is interested, as my title shows, in epithelial sodium channels, or for short, ENaC. And they're kind of an interesting set of ion channels. First, I just want to provide some kind of basic background. They're part of a rather large family of ion channels. The most structurally well-known are the acid-sensing ion channels, or ASIC, primarily found in your central peripheral nervous system, and it's thought to be involved in fear conditioning, pain sensation. And then you have the mechanosensors in C. elegans, the degenerate channels, and when they get, there's some mutations in there where in the pore, where when this channel is constitutively active, it causes worms to essentially die by degeneration. And then you've got the PPK channels that are thought to regulate sodium homeostasis in Drosophila, or insects in general. And then you've got the peptide-gated ion channels in snails. Now, what's really striking about this group of ion channels is that they don't have a common ligand. And so you've got the mechanosensitive ones. Who knows what activates these guys? And then you've got the peptide-gated ones, and then protons. And as my title suggested, ENaC, proteolysis-regulated ion channels. So what's currently understood is that they might be sensitive to proteases, or their primary regulation is through proteases cleaving portions within the extracellular domain that's facing outside the cell, and somehow then they gate or transition from open to close. Another striking thing is that while, for example, ASICs can form homomeric, functionally relevant, physiologically relevant ion channels, homomeric forms in our bodies, what we know so far is that ENaC, this was from the cloning of the genes, and what they've found is that it's most stable when 3 different or homologous subunits. So in this case, we've got the alpha, beta, gamma right here. You see that there's a large macrocurrence, and these recordings are done in oocytes. And when you isolate and just form or express homomeric, or supposedly just the one gene, either alpha, beta, or gamma, you barely see any function. And if you were to just combine it with 2 subunits, it's essentially kind of irrelevant compared to what you see when all 3 subunits are together. And I want to highlight that in order to detect these types of currents for the homomeric or the diheteromeric forms, it requires a lot of RNA and usually days of expression. When you express all 3, you just barely need RNA, and it basically expresses within 12 hours. So you know that it's a preferred assembly. And here it just highlights that ENaCs are, of course, sodium selective. Lithium goes through much more easily. Potassium basically doesn't go through it. And that they are all sensitive to amiloride. So that's the primary pore blocker of these ion channels. The most popular role of ENaC is in the kidneys. In this case, it's just a kind of a little cartoon of the distal nephron. But what it does is It selects or regulates the last 5% of sodium in your body. So essentially decides how much sodium you should have in your body. And a lot of studies have shown that mutation of these ion channels, specifically in the regulatory, there's sections in the cytosolic domain where it will essentially, hyperactivation of these ion channels essentially lead to high blood pressure. And of course, there's the opposite side that reduced activity of these ion channels lead to hypotension, or in some cases, they're called pseudo-hypoaldosteronism, or PHA1. It's kind of a mouthful. But here, what my group wanted to do was understand, first of all, what I want to really kind of emphasize here is that the field itself really does not understand yet exactly how these ion channels work. What they know so far is that they allow sodium to go through. That's like the key thing, and that they're sensitive to amiloride. And of course, their amiloride has all these other derivatives, and that's really kind of solid. And so, but what I want to show here is that, interestingly, like I said, gated by proteolysis, only 2 of the 3 subunits are actually cleaved. So in what we refer to as the alpha subunit, there are identified furin sites. So it's thought that some of these alpha subunits are cleaved inside the cells before they get trafficked out to the plasma membrane. And then we have the gamma subunit, which only has 1 furin site that can also be cleaved while inside the cell. And the proposed mechanism for gamma is that extracellular proteases would then cleave the C-terminal side of that peptide, which allows for perhaps tissue specificity regulation of these ion channels. And what I want to show you here is just an example of human ENaC when exposed to trypsin leads to an increase in activity. And the reason why I'm showing this is I'm going to show you some of our recordings to kind of understand what our channel is doing, or whether or not it's behaving as expected. But as I mentioned before, the most structurally studied cousin acid-sensing ion channel ASIC was solved in 2007, and what it allows us to do basically was it established that these ion channels are trimers. And I'm trying to emphasize this because even now there are still groups that actually still believe that ENaC is a tetramer. And so, but what the ASIC structures and many other subsequent structures after that still show that these ion channels are trimers. But also, what the structure has shown is that if you were to just focus on one subunit, color-coded here, each subunit is assembled like a hand, right? So you've got the knuckle, finger, thumb, you've got the large beta sheets, the palm domain, and then you've got the wrist, and of course, then you've got the forearm. And what's thought or proposed about these ion channels is that perhaps movement of the finger and thumb domain, basically, kind of any conformational changes here, would then be propagated to the TM domains through the wrist region. And that's based on looking at the potential of this ion channel, and what has been identified is that at the subunit interface, there's a pocket that's basically negatively charged. And here, this is the thumb, and you've got portions of the finger here where ideally acidic residues that would be really unfavorable interactions in the presence of low pH, basically high proton concentration, it allows them to be more favorable. So hence, they believe that a lot of the proton sensing is occurring there, and that movement of this particular region is then is the crucial part of the gating of the ion channel. So based on that, what we were thinking is that perhaps the protease-sensitive region of ENaC would be near those primary gating domains. But in order to really understand that, you have to solve the structure. And so turns out it's not so easy. Unlike ASIC, ENaC is not so easy to express. So first, you have 3 different subunits. Second, for some reason, cells are just too smart for these ion channels. And so we had to find ways to basically trick the cells into expressing these ion channels so that we could study them structurally. And so first, to get a homogeneous sample, what we had done was actually mutate all what we thought were the protease sensitive, or the sites that would be cleaved, and basically that would render the extracellular domain completely uncleavable. And second, we also removed large regions of the cytosolic domain so that any floppy domains would be removed. So it's a kind of a classic move. What we could see is that compared to what we would refer to as more wild-type-like, so these are GFP fusions, this would be what we would get if you don't do anything. And this is what the pretty functional, classic ENaC activity. But when you introduce the mutations that I just described, you see that this species is clearly more homogeneous than this terrible expressed wild-type ENaC. But because we had hypermutated the subunits, we couldn't really functionally assay it. And so what we had done was first figure out whether or not identify which subunit we completely killed, essentially. And so what we had done was first combine one subunit with one hypermutated subunit with 2 wild-type subunits. And in this case, just to kind of provide, what it shows is that, yes, when you cleave it with trypsin, you do see an increase of activity. And so what that allowed us to learn is that, yes, each subunit can assemble with a wild-type subunit. It's just, for some reason, they don't go up to the plasma membrane. So then we looked at it in our mammalian cell work and did some immunohistochemistry on it and found that, in fact, what we had done was it just prevented it from getting traffic to the plasma membrane, and that's why we were not able to record from it. Okay, so that's not so bad functionally. And how about, let's try to solve the structure. And it turns out, when you purify ENaC, look at it by cryo-EM on the grid, and basically what you see is blobs, low-res blobs. And immediately, though, one can see that it's similar to ASIC. It's a trimer. Also, I think what's happened, when we saw this, is that the software is not able to distinguish between the extracellular domain and the transmembrane domain, which is at the time was surrounded by my cell. So it wasn't able to align it. And then second, each subunit essentially looks similar. So of course, immediately software couldn't really align that perfectly. So we're not able to see any high resolution features, even just by 2D classifications. And so what we had done was then, of course, went back and used a classic structural biology technique and generate high affinity monoclonal antibodies against ENaC. And so we created these antibodies, or we generated these antibodies, and found that there were two promising ones that allowed to shift, let's say, the ENaC, so indicating that there's a larger complex. And sure enough, what you see is more features, right? So you knew immediately that Fabs allowed for distinct features to come out, and so that really helped align the particles. And so then what I want to then emphasize now is when we generated these monoclonal antibodies, we did not select, or we did not know which subunits they were binding to. So we could not use the subunits, the antibodies to identify which subunit was in the map, like how to assign which subunit goes into the specific density feature. So luckily, and in this case for cryo-EM, glycosylation was our friend. Back in my X-ray days, glycosylation was basically our enemy. But in this case, we immediately identified the beta subunit. So the beta subunit has 11 predicted glycosylation sites. So you knew immediately it was a sugar-coated subunit. And sure enough, when you look at the cryo-EM map, you get these protrusions just kind of coming out of this particular subunit. So we knew that it was the beta subunit. Now, how to distinguish between the alpha and the gamma subunit? We knew immediately that the alpha subunit had a glycosylation site near the wrist region, and the gamma subunit didn't. And that was also pretty clear on the map. And so once we assigned the subunits to whatever position in the map, and also, we validated it by sequence also based on aromatic residues and all the basic residues. Thankfully, we were able then to show, so this is kind of the whole cryo-EM map. And here you've got the beta subunit in red, you've got the alpha subunit in magenta, and then the alpha subunit in blue, and then the gamma subunit in magenta. Why won't it stop? Oh, here we go. So going back to the subunit domain assignments, like in ASIC, each subunit of ENaC is arranged basically like a hand. You've got the knuckle, you've got the finger, beta ball, palm, thumb, but then you've got these protrusions, the blue, here colored in blue. And what you see here is that also when you're looking down the channel down the threefold, what you see is that the subunits are arranged in a counterclockwise manner. So that's viewing from the extracellular side and looking down. You've got the alpha, beta, gamma, and you've got these unique features that are not found in ASIC. And based on that, we decided that we would call it, because we knew that this feature was only unique to ENaC. We knew that this must be the spot for the protease-sensitive peptides or region of the channel. And so we thought that we would name it gating relief of inhibition by proteolysis, or the GRIP domain. We thought that was clever, because when you remove portions of these peptides, it would somehow perhaps change the way it's gripping, right? So it's gripping. And so we thought we would name it that way. And then just to kind of provide a comparison between a proton sensor, here the homomeric ASIC, and now the protease-sensitive ENaC, what you see is that if you just get rid of all the peripheral domains, they essentially kind of look similar. And that's the palm and the beta ball section. But then when you start adding domains in the periphery, you start kind of seeing something a little bit different. And I want to focus on these regions right here. So when you add the finger domains, you immediately see that in ASIC, there's this little loop that kind of is reaching out already into the thumb domain. You don't see that on ENaC. And then you've got this alpha one, which we refer to as alpha one. Here in ENaC, it's starting to make interactions with the thumb domain. In ASIC and ENaC, there are no interactions at all. And if you were to then look at the alpha 2 domain, the alpha 2 goes up, away from the thumb. Here, the alpha 2 is making all the interactions with the thumb. And then you've got the periphery, the grip domain, which of course in ENaC in ASIC it's missing. And so then when you look at it as a whole, if you were to just remove the grip domain, what you see in ASIC is just this pretty cavernous pocket. Here it's filled with what we refer to as the alpha 2. So immediately what you see is that the architecture of the proton sensor in ASIC, proton sensing in ASIC is very different from this proteolytically gated ENaC. And I just want to highlight that the acidic residues that are found in ASIC, here it's all, okay, aromatic residues. And essentially what the protease-sensitive alpha and gamma domains also kind of look similar to the protease-insensitive beta subunit. And here's the cartoon version of it. So that kind of tells you something, that maybe the beta subunit is also doing something, it's just not protease-sensitive. But as any ion channel biophysicist, where's the transmembrane domain? And in this case, it was really poorly resolved, and it's probably because we hypermutated it, also because it was in detergent micelles. So there's a lot of things to be done with ENaC still. So of course, I've already just highlighted it already, so I just want to make sure that I acknowledge people that have done the work on this ENaC structure. Thank you.
Sorry. So we have a couple of minutes for questions, and I'd like to start out. So what is known about how the proteolysis is regulated, how it senses sodium, and is there a abnormal regulation of the proteolysis related to hypertension or pseudo-hypoaldosteronism?
So it turns out, for example, in nephrotic syndrome, it turns out that a lot of the cells actually are hyperactive, their proteases. So there's this evidence that yes, ENaC in the plasma membrane does get hyperactivated or cleaved. And there are peptides of ENaC that get detected in urine, in fact. So there is that, whether the upregulation or increase of expression of the proteases, I don't know how that's regulated, but it seems that that's also kind of coupled together. Now, sodium, well, you asked something about sodium.
How does the proteolysis sense sodium? How does sodium regulate it?
So, well, the sodium part is basically just essentially going through the transmembrane domain itself. As far as whether or not there's too much sodium and what should the cell do, it turns out it's proposed that there is a sodium site in the extracellular domain that upon increase, it inhibits the channel, and if it actually also decreases the likelihood of it getting cleaved. So they call it sodium self-inhibition. So.
Questions? Yeah. Is there a microphone?
Hi, Isabelle. Nice talk. I'm just curious about the observation with trypsin, so the proteolysis. As I understand it, I think you're sort of suggesting it's relieving some sort of tonic inhibition on the channel.
Yes.
Right? What's the evidence for that? Why do you think there is tonic inhibition? Because if you look at the oocyte recordings, the response is fairly well maintained. So something that happens very rapidly, or couldn't it just be that you're potentiating the open probability of the channel independently of inhibition?
So that's a good question. There are thoughts also that lipids themselves are actually regulating the activity in the ion channel that's independent of the extracellular domain. So how is that coupled? That's not exactly known. That's why I wanted to kind of emphasize that the field itself is not completely set yet on how it's if it should be regulated.
We have another question back here.
Hey, good morning. Is there a chance that the proteolysis could be relevant to cystic fibrosis, where the channel's upregulated?
I'm hesitant to say it, mostly because there's still kind of two, or not two, but there's still, it's this ENaC role in CF is still not completely solid in a way. So I'm not familiar with the cystic fibrosis or ENaC regulation in cystic fibrosis at all, so I can't say very much. The only thing that I could say, I suppose, is the acidity that occurs in the lungs, for example. It's thought that human ENaC is actually sensitive to pH. So it can be hyperactivated when the pH gets lowered, which in the case of CF, I think that does happen.
One more question, anybody? Going once, going twice? Thank you very much for a beautiful talk.